I guess I'll start right now. The Andrew Connor Curtis Endowed Lecture Series for Tumors was established about 6 years ago in 2016 from a generous gift from Andrew Kovacs with the objective of showcasing advances in the field of neurosurgery and neuro-oncology. And it's really featured a wide variety of distinguished visiting professors, including Eric Holland, Tracy Batchelor, Hugh Defoe, Fred Lang, and Gallery Zada. This year, we're extremely fortunate to have Dr. Amy Heimberger from Northwestern as our distinguished brain tumor visiting professor. We were supposed to, unfortunately, with I guess weather delays, we're doing this via Zoom, but we're still lucky enough to be able to hear from her today. So, just to give you a little bit of background on Dr. Heimberger, she completed her medical school at WashU in St. Louis and finished her residency and fellowship at Duke. Then she transitioned to faculty at MD Anderson for about 18 years, and now just recently made her transition a few years ago, I believe, to Northwestern. She's one of the most distinguished neurosurgeon scientists in the country, really revolutionizing the field of glioma immunotherapy. She's principal investigator on 3 R01s and a senior author on several papers in prestigious journals like Cancer Cell, Nature Medicine, Clinical Cancer Research, Journal of Clinical Investigation, Nature Genetics, and many more. We're extremely lucky to have her present this morning, and I'm hoping we can learn a little bit from her model of becoming a neurosurgeon scientist. So the first lecture, I think, is on basic science and kind of translational work, and then the second lecture is on kind of how, for the residents, kind of looking at her model and becoming a neurosurgeon scientist. So thank you again, Dr. Heimberger, for coming.
Oh, thank you very much. Again, my apologies that I'm not able to join everyone in person. I would have very much liked to. I really love the city of Miami. It's so dynamic and fun. And of course, you got the beach right there. So lots of wonderful things. And I have several good friends that are in Miami as well. So I'm very disappointed I couldn't be there in person. I believe this is a CME event. If not, I apologize for showing you the conflict of interest slide. I will talk about a couple of things in which I might have a perceived conflict of interest. I'm going to talk a little bit about a STAT3 inhibitor, which has been licensed to moleculine. And I'm also going to be talking a little bit about a brand new compound that was developed just a couple of years ago called a STING agonist that has also been licensed to a company called Immunogenesis out of Houston. So today I thought I would focus a little bit on a sort of a patient-related matter, which is the paradox of why immune checkpoint inhibitors, and when I say immune checkpoint inhibitors, I'm talking about things like, anti-PD-1, anti-PD-L1, et cetera, why it seems that they have either radiographic response or an increase in survival in certain patients, whereas they seem to fail in the vast majority of glioblastoma patients. Really, in many ways, immunotherapy revolutionized the field for melanoma. When I started out, the median survival for stage 4 melanoma with brain metastasis was under 6 months, and really many sort of pivotal studies, the immune checkpoint inhibitors as well as BRAF and MEK inhibitors, have really sort of pushed out that survival for melanoma, and its prognosis is not as dire. Unfortunately, the same cannot be said for glioblastoma, and there's been a number of high-profile phase 3 clinical trials where, for the vast majority of glioblastoma patients, these types of therapies do not have a meaningful benefit. So one of the things that I was taught initially in medical school was that the central nervous system is immune privileged, and sometimes people still bring that up as a potential notion. I did show you a clinical study in New England Journal of Medicine on the previous slide that there are radiographic responses in the central nervous system for melanoma metastases. But as surgeons, usually you are visual creatures, and so I thought I'd produce evidence visually for you to the fact that there is immunological recognition within the brain. These are mice models that I'm going to show you. So what we do is we create a brain window in the calvaria. In other words, we drill it down with a little tiny, almost like a little Dressel tool. And then we put a glass plate over the defect in the bone. And then these mice have actually been genetically modified in such a way that the T cells, the CD2 positive T cells, are shown up as red. And then CD11C presumptive dendritic cells show up as white in these particular video clips. And what you're doing is you're looking at a live mouse directly into the central nervous system. You can see the cyan blue, those are the blood vessels. And then what we do is we implant GL261, which is a mouse syngeneic glioma cell line, into the artery of the mouse, and then those go up the vessel and then marginate out and then situate into the parenchyma of the brain. And the reason why is we don't do direct intratumoral deposition of the tumor is that can induce an intrinsic inflammatory response in of itself. So on the video on the left, you can see this GL261, this cobalt cell sitting right here in the brain. You can see these CD11C dendritic cells, they're reaching out, they're touching, they're sort of interrogating and sensing the microenvironment. And then these T cells you can see are scurrying about. And this is day 2 after we've actually done the carotid injection. And then I want you to take this GL261. We're actually going to magnify that up, and we're going to look at this a couple days later, specifically day 7. Now you can see that that dendritic cell is directly touching, making physical contact with that glioma cell. And now the T cells have actually found that glioma, and then imaging a day or 2 later, that glioma cell is gone. So it really does indicate that there is a propensity, or an ability, I should say, for the immune system to recognize cancers within the central nervous system. And now the rest of the talk is really going to be about, well, what are the key issues that limit this, and why does this not happen in the context of CNS tumors like gliomas relative to metastasis? Okay, so key fundamental paper in the field, one of the ones that I think are most pivotal to sort of getting to the bottom of at least one of the issues, is this beautiful study done by Becker. It's published in Cell 2 years ago, where they did single cell mapping looking at the immune composition within tumors. Specifically, they compared and contrast gliomas versus metastasis. And this paper showed a number of key findings. Number one, they show that there's a difference in frequency and the number of leukocytes, in other words, bone marrow derived immune cells, that infiltrate into brain metastases relative to intrinsic gliomas. They also found that there's, many times we think that monocytes are just a single population. In fact, there's probably subgroups of monocytes. And what they showed was that tumor lineage or types actually influence the differentiation states of those monocytes. And I suspect probably the various types of monocytes that actually are in that microenvironment. They also showed that brain metastases harbor a higher frequency of regulatory T cells, and that you see both activation and exhaustion in the lymphocytes from the metastatic tumor niche relative to glioma. And the reason that this is important is that these T cells can become so exhausted that they can't be reinvigorated with immune checkpoint inhibitors. If they're partially exhausted, in other words, if they still have markers of activation and maybe some markers of exhaustion, then they have the ability of potentially being resurrected. Most of the T cells within glioblastomas, and this was done by Peter Fecci at Duke, really show this profound terminally exhausted state. In other words, they're dead as doornails. No matter how much of this stuff you give them, they're really not going to come back and be activated. So that's one or two of the differences. Another key difference is the types of immune surveillance between cancers. Now, I want to clarify that this is systemic malignancies that I'm showing you on this panel of analysis. And this was actually CyTOF data. And we profiled, at pan cancer, we profiled GBM relative to non-small cell lung cancer, renal cell carcinoma, colorectal cancer, and pancreatic cancer. And right now on that top panel, what we're looking at is just frequency of immune cells. And there's a relative paucity of CD3 cells, and that includes the CD4 helper cells and CD8 cells in GBM relative to other cancers. And then you see a relative increase or a relative high frequency of CD68 monocytes. And these give rise to your tissue-derived macrophages, et cetera. The other thing that's different is the immunosuppressive mechanisms that are utilized between GBM and other types of malignancies. So PD-1 and PD-L1 are very frequent for melanoma, lung cancer, as far as their expression. But we've done a series of papers that show, at least for PD-L1, that's relatively rare in glioblastoma. And additionally, GBM, as we all know, is very heterogeneous. And it utilizes a wide variety of different immunosuppressive programs. So it could use a lot of TGF-beta, IL-10, in other words, cytokines. Some tumors have higher degrees of T regulatory cells. And then the immunosuppressive pathways that it uses is different as well. So this was one of the things that we found with the CyTOF, which was that there was a relative higher frequency of CD73 pathway components in glioblastoma. And I'm going to talk on that in just a moment. If you genetically ablate CD73 in the background of mice, you can actually get immune checkpoint inhibitors to work in GL261, and I'm showing that in the bottom panel here. Of course, genetic ablation is always the best case scenario, and therapeutics never quite approximate that sort of gold standard that you see in that particular context. So one of the things we did was we were very interested in helping our neuro-oncology colleagues in identifying immunotherapies that might actually have real applications for GBM patients. And so we started this profiling project where, as a surgeon, we take out the tumor and we immunologically profile the immune cells. We also looked at the peripheral blood, and the key was to try to prioritize which types of immune therapeutics that we wanted to move forward. And so we only scanned for targets that we had available therapeutics at the time. Now, what I've done is I've converted very complex flow analysis cytometry data into a heat map, because there's no way you can look at 1,000 flow cytometry diagrams and sort of assimilate what the story is or what the bottom line is. So when you look at this heat map, as you go to the hot pink color, that means it's the most frequent, or you see a higher incidence of that particular target on the immune cells. Certainly you see PD-1 expressed on CD8 and CD4 tumor infiltrating lymphocytes on the left series of panels. But you'll notice that what you really see most commonly is the adenosine receptor. And we also see that on the innate immune cells as well. And so this is a pathway, it's the adenosine CD39 CD73 pathway. I just touched on it on the previous slide, where what happens is if you have CD39, and it can be expressed either on the tumor cell or on the immune cells, it actually initiates ATP conversion to AMP. And then if you have CD73, and it can be expressed in the tumor cells or the immune cells in the tumor microenvironment, it converts AMP to adenosine in the microenvironment. And then adenosine binds to the adenosine receptor, which is expressed on both the T cell compartment as well as the innate compartment. And when this happens, what the T cell does is it essentially becomes unactivated. So if it had an effector response, a killer effect, then the adenosine engagement on the receptor turns it off. In the context of macrophages, when the adenosine engages the receptor, it actually makes the macrophage more immunosuppressive. So in many ways, this is really a very sort of key hub for mediating multi-immune compartment immunosuppression. So both T cells and macrophages. So based on what I'm telling you right now, you can obviously see where we would be going with this, which is we would start pulling available compounds and start testing them preclinically to see if we have a signal for moving forward with the adenosine pathway inhibitors. And when we actually perform these experiments, and I'm showing you only a limited number of experiments, we sliced and diced this dozens of different ways. But it doesn't really matter whether you use a blood-brain barrier penetrant adenosine receptor inhibitor as a monotherapy, which I'm showing in the upper left panel, whether you start moving that together with things like immune checkpoint inhibitors to try to maybe have an impact on redundant immunosuppressive mechanisms, whether you start using all the components of the adenosine pathway as far as inhibitors all along that pathway, the result is essentially the same, which is that even though there might be a statistical significance, it really isn't biologically compelling. So whenever I see in vivo experiments like this, I don't usually use this as a signal for us to move forward with implementation into human subjects. So the question is why is this not working? So we did some proof of principle experiments, which I'm showing you in the bottom panel here. So what we did was we set up an assay. We activated T cells, we turned them on, and then essentially we blocked the adenosine pathway with a compound called NECA. And then we added our compounds back in to see if these compounds were working the way they're supposed to be. And you can actually see that when I add in adenosine receptor inhibitors, that it restores the interferon function in the CD8 T cells. So these compounds work perfectly well. There's nothing wrong with the compound per se. But if you add in either glioma supernatants or you do any kind of sort of co-culture with glioma cells, essentially these compounds do not restore the activity of those T cells. So even though the initial phenomenon of T cell exhaustion was described in the context of things like anti-PD-1, I suspect this is going to be universal. In other words, none of the compounds, I suspect, will be able to reverse T cell exhaustion. So we really need to start thinking about therapeutic modalities that increase the T cell frequency into the tumor microenvironment and increase the number of activated T cells in the tumor microenvironment moving forward. Now, one of the things neurosurgeons are particularly good at, obviously, is surgery. And we have a key and pivotal role to play in neuro-oncology clinical trials. Now, Ingo Mellenkopf has done some of the initial studies where we evaluated this sort of strategy and targeted therapeutic approaches, things like EGFR inhibitors. But increasingly, you're going to see more and more of sort of these window of opportunity clinical trials utilized in the context of IO, in other words, immuno-oncology agents. And there's a number of compelling reasons for seeing this and neurosurgeons being increasingly involved in these types of clinical trials. There's 4 key steps you need for immune activation and clearance of cancer. You got to have a target, you got to have activation, you have to have sufficient trafficking, and you have to have maintenance of an effector response. If you're just monitoring peripheral blood, you may not necessarily be able to interrogate the trafficking of your immune response into the tumor microenvironment, and you certainly don't know whether you have maintenance of your effector response in the tumor microenvironment. So the reason to do these window of opportunity studies in immuno-oncology specifically is, number one, you can verify if you've got an anti-tumor immune response. In other words, number of T cells there, are they still activated? Do you see direct cytolytic killing? I want to warn you, frequency does not mean function. So just because you got a CD8 T cell there or CD4 T cell there, it doesn't necessarily mean they're doing anything. Increasingly, we're starting to learn that there's going to be key distribution limitations for immune cells in the tumor microenvironment. If you've got an IO agent, you want to look at, okay, what's the concentration that's achieved within the tumor or the adjacent infiltrating area. You can do PK analysis of these agents. And specifically in the context of what we've done so far, utilizing this particular clinical trial strategy, is we're using it a lot as a go, no go for later stage clinical trials. So before you commit 10 million plus to a phase 2 clinical trial, if you're not achieving what you think you should be achieving in the tumor microenvironment, then perhaps you need to go back to the drawing board and either rethink the combination or do additional preclinical analysis, et cetera, or refine your therapeutic. So there's 3 ways of doing this window of opportunity clinical trial strategy. Number 1, you can biopsy the tumor, and then you can give a drug or an agent or a therapeutic or cellular therapeutic, and then you can take the tumor out and then compare between the 2. The second strategy is you can randomize patients. You have one set of patients that get the therapeutic, another gets either nothing or standard of care. And then you compare the microenvironments between these 2 cohorts. And then the third way you can do these clinical trials is you can have a patient recurrence, perhaps. They get the drug, you take out the tumor, and then you compare that relative to tissue bank. And there's inherent sort of disadvantages and challenges and advantages to all 3 of these. And if you wish, we can certainly discuss that in the last 15 minutes of questions in more detail. So one of the first ones that was kind of done this way was a study that I did with Dr. John DeGroot, who's now at University of California, San Francisco. And we took recurrent GBM patients. And the purpose of this clinical trial was really to investigate what's happening in the tumor microenvironment. And so these patients were treated with anti-PD-1, Pembro. And so they received 2 doses of that drug. And then we took them to surgery. They also had their peripheral blood monitored as well. And then they subsequently were treated after the resections with the pembro until they subsequently progressed. The progression-free survival on these 15 subjects was 5 months. Overall survival was 21 months. Now, if you're an optimist, you would say that this is a clinical response. And if you benchmark it, let's say, to New England Journal of Medicine, Stoop article, etc. However, patients that get referred to high-performing centers, including places like yourself, Miami, MD Anderson, Memorial, etc., usually, if you go back and look at the patients retrospectively and historical, those folks usually do better than those that are seen, obviously, out in community practice. So if you benchmark this analysis relative to that sort of historical cohort, then it's not necessarily showing you a signal of response. Now, regardless of how you interpret the PFS or the OS of this particular clinical trial, the key factor here is that the overwhelming data really indicates that there's not much of an effective response at the time point that was measured. So on the top, what you're seeing is CyTOF data. CD45 is a marker of the entire immune population. And please notice that the overall CD3 T cell composition is extremely small, especially relative to things like the CD68. All the other blue cells you're seeing, those are tumor cells. So when you do immunohistochemistry and start doing a more sort of deep dive in the functionality of those T cells in the tumor microenvironment, they really do lack things like interferon, granzyme B, perforin, etc. So although there's a couple that are scattered there, they're really not effectors. And I will tell you as an immunologist that I find this sort of profiling completely underwhelming with regards to an anti-tumor immune response. So sometimes I see slides, on national presentations where they show me 3 CD3 T cells, and I know that this is where they've got the most greatest density, and that's very underwhelming. It's not convincing to the immunologists in the crowd. When we started doing a deep dive into the predominant population within the tumor microenvironment, those are the CD68 monocyte cells, you'll see that almost all of them are inhibitory, as denoted by the red. So you'll look at their phenotype, even though they might be expressing things like MHC, and HLA, you'll see HLA, DR positive, etc. You'll see that there's a lot of immunosuppressive markers that are also co-expressed on these cells. Intriguingly, these cells also express PD-1. And folks a lot of times think of PD-1 being exclusive on the T cell, and that's not true. So Irv Weissman, who's an immunologist out in California, he initially described PD-1 being on macrophages, and PD-1 engagement on macrophages blocks their phagocytic activity. Thus, they're the first couple steps needed for them to be antigen-presenting cells. But we also did some modeling in Eric Holland's genetically engineering mice models. Now, this was done with Ganesh Rao, he's chair down at Baylor. And we took Eric's gems and we actually knocked out the CD8 T cell population. And then we actually treated those mice with anti-PD-1. And Ganesh and myself had this debate of whether this was going to ablate the therapeutic effect in these mice. So on the left, this is a Kaplan-Meier curve showing you that you have an increase in survival when you treat these mice with anti-PD-1. And you also have an increase in survival with anti-PD-1 when there's no CD8 T cell. So this was a little bit of a paradox, but I've already kind of given you a hint as far as why this may be working. And when we looked at alternative immune populations that can have direct tumor effects, those include things like CD4 cytotoxic cells. Those are uncommon, but it does happen in cancer. We never found those. We looked in the peripheral blood, we looked in the tumor microenvironment. We also looked at NK cells. And again, we didn't see any NK cells really in the tumor microenvironment that would suggest that they were capable of direct sort of cytolytic activity against the tumor. And then we ultimately profiled with a multi-gene panel what was going on with regards to the macrophages. And what we found was there was really a very profound gene modification or I would say, push towards what's known as the M1 macrophage. And so these are pro-inflammatory, anti-tumor macrophages, and these are considered highly desirable from an immunological perspective. So the question is okay, M1 macrophages, those cells can elaborate things like iNOS and other factors that can directly kill tumor cells. And we became a little sort of intrigued with the notion of microglia, and are they also contributing to the biological sort of increase in survival we're seeing here in these CD8 knockout cells? And so to sort of start to address that, and it's not completely resolved at this junction, and additional investigation is necessary. Some work that we did with Pravesh Gupta, he was trained as an immunologist in Singapore. He did some single cell RNA sequencing, and this is a lot of data, but I want to just illuminate a couple little points here. I've always been taught that microglia, or I thought that microglia were a single cell population. That's probably not the case. There's probably a diversity of microglia populations, especially in their transcriptomic profiles. And when you actually start doing things like gene ontology, you can see that there's a great deal of heterogeneity in the microglia population for human subjects, and this is gliomas, as defined by the markers of CX3CR1 and P2RY12 positivity. Some of these microglia have antigen presentation and phagocytic capabilities. Some of them are pleuroinflammatory, but there's subsets of these microglia that clearly do not have an immunological sort of reactive role, as you can see from the gene ontology. But what I also thought was quite interesting is that you see subsets of microglia that have direct tumoricidal activity, at least denoted by their gene profiling. In other words, like things like lysozyme and granzyme B, et cetera. So this remains to be further elucidated, and it requires immune functional assays now at this point to actually validate these kinds of findings. This does, however, provide us with a contextual way to move forward with immuno-oncology for GBM. And I'm going to do two sides of how to control macrophages. The first side I'm going to talk about is how to turn on the macrophage to elicit a T cell response. And then at the very end, I'm going to talk about how to modulate the brakes of these cells. Remember, these are the dominant immune cells within the tumor microenvironment. So the CGAS STING pathway, you guys are seeing more and more papers on this particular pathway. It's a very hot area right now in immuno-oncology. Essentially what happens is you have a tumor cell, it may undergo necrosis, apoptosis, whatever, and it may actually have pieces of it that are taken up via phagocytosis with your macrophage or microglia. This subsequently then generates sort of these double stranded DNA. They're in the cytoplasm of the macrophage that then engages CGAS. You have a signaling cascade that then subsequently triggers STING, and then STING modulates transcriptional activation of things like interferon and TNF alpha, and that's pro-inflammatory, and that's very good. The CGAS and the STING pathway also enable the loading of antigen onto MHC for antigen presentation. So it's pivotal for that process as well. So think of CGAS STING as essentially your accelerator on the car. In contrast, your brake is the STAT3 pathway for these cells. So you can have reactive astrocytes. They elaborate things like IL-6. IL-6 then engages the IL-6 receptor on ligand engagement. You get tyrosine 705, phosphorylation, STAT3 dimerizes. It then goes into the nuclear compartment, and then it essentially triggers transcriptionally immunosuppressive cytokines, things like TGF-beta, IL-10. When STAT3 gets triggered, essentially now it then comes back around and it stops antigen presentation. And that's how come you see these big black or these big red X's essentially saying, no, this no longer happens. That's when you actually trigger the STAT3 pathway. So therapeutically moving forward, one of the key elements is you could actually engage STING, CGAS to turn on these cells, or you can block STAT3 as a brake In other words, you stop the brake on the macrophage. So one of the compounds we developed with Michael Curran, he's an immunologist. This is what I was alluding to at the very beginning of the talk. We worked on two compounds. They're very, very similar. They're denoted as IX8779 and IX8803. There's publications of the description of how they were developed. You just have to PubMed it. And these compounds are very closely related. They're almost identical, but there's some subtleties between them. I'm showing you on the left the proof of principle engagement of both 8803 and 8779, these STING agonists. Both of these very nicely engage and enhance NF-kappa B activation, which is downstream from the pathway, and ERK3. And additionally, when you inject these into tumors, into the tumor microenvironment, you see nice proliferation of CD8 T cells. You can see activation of those cells. And you can see downregulation of things like CD206 and arginase. These are markers of M2 or immunosuppressive macrophages. And you see the upregulation of things like stimulatory molecules like CD80, CD86. In a humanized model of U87, so these are mice that have been reconstituted with CD34 hematopoietic human stem cells. So the immunological response is human. You can see that there's an increase in survival in these mice that have an orthotopically implanted U87. And these mice were treated on day 5, 10, and 15 with just 5 micrograms into the tumor. And then additionally, we tested the standard models, things like GL261 and CT2A. But for the sake of time, I'm going to show these two models that have just had some data that have come out on them in JCI Insight. These were initially derived from mice we described in Nature Genetics with Jen Flaw. And these particular gliomas are immune checkpoint resistant. So recapitulating in some ways the biology that you see in human subjects, they have a low tumor mutational burden, whereas GL261 has PD-L1 expression, higher tumor mutational burden relative to these particular cell lines. In both of these that are resistant to immune checkpoint blockade, PD-1, CTLA-4, et cetera, you can see that there is a biologically compelling increase in survival in QPP4 as well as in the QPP8. Now, the orange line for 8879, you can't see in the bottom panel because essentially it's 100% survival. So it's directly underneath the purple line in that bottom right-hand panel. Now, this is direct intratumoral injection. And so one of the key questions is, do you need to bathe the tumor microenvironment as a surgeon in order to deliver this therapeutic? So in other words, do you need to think about convection enhanced delivery, et cetera? So we did a series of these scopal experiments where what we do is we take a mouse and we implant a tumor on both hips of the mouse, but we only inject one side. In other words, the contralateral side is never treated. And in the blue, you can see that the mice are treated on the left and the right, and you can see the suppression even on the contralateral untreated side. In other words, on that top row, you can see the right flank, that's the one that got injected. We did that for prostate cancer, and we also did that for melanoma, where you can see, again, the contralateral side that is not injected. You can see that there is growth suppression in these mice. So clearly mouse models are little itty bitty tiny tumors. And so we then worked with our veterinary colleagues, and this was specifically at Texas A&M, where we identified dogs. Usually they presented with seizures. They had contrast enhancing masses on MRI scan. This is one of these cases. And what we did was, and you can see the device that we worked with the neurosurgeons at Texas A&M for injecting out. Clearly, we don't need something like this. We've got much more sophisticated equipment we use on a daily basis for our patients. But neurosurgeons, surprisingly, doggy neurosurgeons don't do resections for glioblastoma. They may sometimes do it for things like meningiomas, for dachshunds, that kind of things. So we actually had to develop some of the equipment for the vet staff. And so here you can see the device where we actually injected the STING agonist. This on the right side are MRI images. Please look at the middle column. After the first injection, you can see that there was a marked radiographic response. We injected the dog again, and you can see we had a complete radiographic response. And this particular dog received 2 doses at 15 micrograms, so a very small dose. So this was our go signal for moving it into a clinical trial. So we have multiple preclinical models that show therapeutic effect, as well as a large animal model. And this is how we prefer to actually vet things, that we invest in moving them into clinical trials before committing large amounts of patients and funds towards them. One of the things we have been working with David Ashley, who's at Duke, is trying to understand which patients may be more predisposed to responding to STING. STING actually becomes deactivated in the tumor cells. And so we published this data in Cancer Cell two months ago, where certain gliomas and certain cancers, there's a methylation site in the promoter where this actually gets turned off. If you have a cancer that has a lot of high tumor purity, in other words, very low macrophage infiltration, we would actually suspect that this drug would not work particularly well. And this actually was borne out in some in vitro assays that we did. We took newly diagnosed GBM tissues, we placed it in culture. We then added in STING agonists, and you can see you get a very nice, robust induction of things like CXCL10, which is really important because it's a key chemokine for T cell infiltration into the tumor. However, if you do that exact same experiment and you get rid of your CD14 monocyte macrophage population, you lose the elaboration of all of those good pro-inflammatory cytokines. So the way to think of this is we want a dirty tumor moving forward. In other words, we want a tumor that has macrophages in it, because that doesn't become, the STING doesn't become deactivated in those cells, only the tumor cells. So we've actually developed an assay that we can do via a stereotactic biopsy, where we can look at tumor purity, and this is based on DNA methylation profiling, where we can simultaneously look at the STING methylation promoter, as well as ascertaining the myeloid density. So this is the way it's actually incorporated into a proposed phase 1 clinical trial that we're hoping to get launched in a year or two. We're still working on the IND of this particular compound. Cohort 1 would be a recurrent GBM patient cohort. We've also developed a PET imaging strategy where we can look at STING engagement in preclinical mice. So we're planning on imaging the patients before we deliver the drug. The surgeons will come in and biopsy. The purpose of the biopsy is to get the methylation status and the tumor purity score. The same biopsy needle trajectory will then introduce the STING. We are also going to be monitoring CSF. I'm specifically very interested in looking at CXCL10, and it's been previously shown that it can be detected in CSF. And then we'll re-image these patients with that STING PET imaging. And then for those patients that did not get, let's say, a good surgery the first time around, and it happens sometimes for a variety of reasons, or let's say they clearly would benefit from recurrent surgery, we would obviously take that cohort 2 patient, and those would be the ones that would be profiled through the window of opportunity assessment. And we'd be specifically looking at things like T cell effector function in those patients. Do we see reprogramming of the macrophages? Do we see antigen presentation occurring in the tumor microenvironment? Many of those things would be assayed in that particular cohort. And again, if we do not see the desired therapeutic or immunological responses, then we're not going to advance it to later stage clinical trials. So distribution is important. I wanted to show this particular clinical trial. This was another study we did with a window of opportunity analysis. In other words, another clinical trial. The neurosurgeons were pivotal in really sort of identifying therapeutic response or not. Some of you may recall that there's been a number of clinical trials done with dendritic cells where they pulse on them CMV, PP65, and other sort of antigens. I'll bet there's small numbers of patients on those clinical trials. It did look like at least that there was some sort of therapeutic response potentially in those studies. And so we wanted to actually see if we could validate or duplicate. So as opposed to dendritic cells, we directly took the patient's own autologous T cells. We grew them up in culture. We activated them. In other words, we expanded out the T cells that were not completely exhausted. We obtained them from the peripheral blood. And then we gave them back to the patients. The way we allow for clonogenic expansion in this particular cohort study was with dose-dense temozolomide. There's other ways you can do this. So these patients were recurrent. They underwent a leukapheresis. We manufactured the CMV specific T cells. While we were doing that, they got dose-dense temozolomide. And then we infused them with multiple cycles of these CMV specific T cells. Progression-free survival in this clinical trial was 1.3 months. Overall survival was 12 months, small cohort of subjects. We would not be advancing this for the reasons I'm going to show you. I think clearly most people on the line would agree that this doesn't show much of a clinical response. However, there was radiographic responses when we measured volumetrically the contrast enhancing areas on these patients. And the oncologists on the call will realize that patients that are MGM methylated can respond to dose-dense temozolomide radiographically. So this was the confounder for that possible positive signal radiographically. We then did extensive immune monitoring, and although you see an increase with each cycle in these cohorts of patients, in other words, there was an increased frequency of the CMV specific T cells, as there should be because we're adoptively transferring these T cells. The thing that became disturbing was the fact that these T cells did not look particularly activated to us immunologically. So you're not seeing markers of activation like CD107A, you're not seeing TNF alpha, you're not seeing gamma interferon. And one could certainly argue, well, maybe they're marginating into the tumor microenvironment. So as a surgeon, we went after that. So window of opportunity. And when you actually infuse the patient with these CMV specific T cells and then pull them back out, they're essentially dead as a doornail. So they're not elaborating things like TNF alpha. They're not producing interferon. They're not activated. But what's even more disturbing is I want to show you this immunohistochemistry image over here on the right. So this is a marker that picks up CMV specific T cells by the brown staining. This is a blood vessel. Here are RBCs in that blood vessel. This is GBM. And what you'll notice is I'm only showing you where the CMV positive specific T cells are. And notice they're right here just in this perivascular space. So increasingly, the data is starting to emerge that T cells do not uniformly distribute throughout the tumor microenvironment. And this is where it becomes really important for you as a neurosurgeon to actually be involved probably in therapeutic delivery. So there's a variety of different strategies for doing this. At Northwestern, what we're doing with Adam Sonnabend and Roger is low intensity pulse ultrasound. This is a strategy that can supposedly open tight junctions, induce transcytosis, can open up transendothelial fenestrations. And so we were very curious of whether it can improve the distribution of things like immunotherapeutics. And of course, other people are interested in it from the perspective of opening the therapeutic compendium for most chemotherapeutic agents, since they do not usually have blood-brain barrier penetration. This is the device we use preclinically. So up here, this is the little platform. In the center is a little water fountain, and the ultrasound is transmitted through the calvary of a mouse because it's so thin, utilizing pulse ultrasound. And so the mouse gets lined up. The mouse is anesthetized. You have to shave the mouse head so that you have nice contact between the ultrasound as well as directly to the skin, and there's equal distribution. And then you do a tail vein injection where you give microbubbles. So the ultrasound, what it does is essentially, it allows those microbubbles sonoview that is FDA approved to essentially open up some of those little blood vessels. And so it allows the extravasation of whatever you're giving. And here's proof of principle showing you with Evans blue, the areas that we were sonicating, which was near the area ultimately where we implant tumors. So we wanted to make sure our therapeutic strategy preclinically was working adequately. So one of the things we wanted to do was test anti-PD-1, and you'll be like, but it works on T cells. Well, remember earlier I showed you data that, that it probably also works on macrophages. It may be also necessary for microglia in the tumor microenvironment, et cetera. So we were very interested to see whether this might enhance anti-PD-1 therapeutic effect. So we labeled PD-1 and actually sonicated. These are non-tumor bearing animals here, so intact blood-brain barrier. And you can see the signal, the bioluminescent signal increases pretty markedly with multiple sonications. And then we implanted GL261. We started giving PD-1. This is very early when there's microscopic tumor. The paper that's published shows you at later stage clinical trials as well, or later stages of tumor initiation, that there's an increase in survival with the sonication. And there's some induced immunological memory here. This is with CAR T cells. There's been a number of CAR T cells that have been advanced in the clinical trials for glioblastoma patients. This is a bioluminescent EGFR CAR in mice. You can see the increased sonication signal. And you can see an increase in the signal over time. And this is immunohistochemistry showing the persistence of the T cells in the microenvironment utilizing blood-brain barrier opening ultrasound. Please notice in this immunohistochemistry image that I'm not near a blood vessel, that I'm in the actual tumor itself here with an increased density of T cells persisting. This is the survival when you give CARs, and we give these IV. And when you add in the ultrasound, there is an increase in survival. You can also deposit things like antigen presenting cells into the tumor microenvironment, which can draw in T cells. And this is a strategy that was not even optimized yet for dose and schedule, showing you proof of principle that you can certainly utilize the ultrasound in this manner. So as a neurosurgeon, one of the things we obviously do is take out tumors. I typically like to do these oncologically or in block if possible, and clinically, if it's obviously in the interest of the patient to do so. We wouldn't do something like this. Obviously, it's something in the motor strip. What we do is we do in block, and then we actually prepare a wedge. This wedge allows us to interrogate the tumor microenvironment in continuity. And so we've actually analyzed a number of specimens in this way. So in the bottom right-hand panel, you're seeing the stains for each slide I'm showing you. So the nuclei is going to show up as dark blue. CD3 T cells are your turquoise. Phosphostat-3, remember I told you that's the brake on the immune system, is green, and it's going to be in the nucleus. CD68 is going to be your monocytes. GFAP is going to be your tumor cells or your astrocytes. And then CD163 is a marker of immunosuppressive macrophages. And on some slide, I'm also going to show you some dendritic cells as well with a marker of CD11C. So on the right, this is the necrotic core. You can see as you start moving through the tumor, and then you start getting to the infiltrating edge. And the immune surveillance is markedly different in each of these zones. So here's the infiltrating edge. You can see that you got a lot of sort of CD163 cells. In the tumor itself, there's a lot of CD68 monocytes. And then when you get in areas of necrosis, you see a lot of CD163 macrophages, and every once in a while, rare little isolated T cells, but they're very rare and unusual in the area of necrosis. Mostly it's just all macrophages. This is what reactive brain looks like utilizing this multiplex imaging. Here you can see a reactive astrocyte. This is GFAP positivity. Here you can see that very marked nuclear phosphostat-3 expression. Microglia have CD11C and CD68 co-expression, and here's actually microglia. You can sort of see the amoeboid processes. One of the key issues is many banking facilities, what they do is they enrich for tumors. And One of the key issues is that you might be missing some very important immunological information when you do tissue bank analysis. So here's what your pathologist sees with the routine H&E. This is a low-grade glioma. Here's the glioma on the right side. Here's the interface. Gliotic plane, a lot of times is what we call it. This is the brain over here on the left side. This image here is on multiplex. So this is the exact same image as this. So you can see in the tumor, lots of CD68 monocytes. Right at this edge, you'll see where there's quite a bit of T cells. None of these T cells have phosphostat-3 in their nucleus, which would imply that they're not immunosuppressed. But you need additional markers to determine whether they're actually capable of any kind of effector function. And then over here on the left side, this is the brain. You'll notice there's quite a few CD163 cells. This is what a blood vessel looks like in GBM. So here's the blood vessel. You can see the RBCs in the center. The blood wall is lined with CD163 macrophages, which means as soon as a T cell tries to get access into that tumor microenvironment, the first thing it actually comes up against are these immunosuppressive macrophages. So there's really a profound wall of sort of immunosuppression at the very beginning before it even can get underway. And I'm not sure how well this is transmitting via Zoom, but the nuclei here in these CD3 positive T cells is actually green. So they're probably either Tregs or they're profoundly immunosuppressed. This is where these T cells hang out. And this is standard of care treated GBM patient. So this is not an immunotherapy treated subject. So again, I'm showing you the same observation, which is where the T cells like to hang out. In Mets, T cells like to hang out in the stroma. And so here's metastasis right here, and here you can see all the T cells. When you just biopsy across these various zones, you're probably missing some pivotal information. So this is, I really like this slide a lot. Here's a metastasis. We flipped out one of the markers for cytokeratin stain. And here you can see that there's a gradient of density here with these macrophages. So it's very high as you get close to the metastasis. And as you move further and further away from this metastasis towards normal brain, you'll see that the density actually goes down. So that means there's something in these cancers that's really sort of pulling these macrophages sort of in the adjacent brain towards it as a way to protect it from immunological reactivity. One of the key things that's kind of interesting is that there's differences in how these cells interact. And increasingly, you're going to see a field of interactome analysis that's starting to emerge. And this is not just specific for gliomas, but you're going to see it in cancer. When you look at the edge of metastases, you start seeing cluster interactions. And these cluster interactions are where a big group of T cells or immune cells or macrophages are getting together and are interacting. You see these cluster interactions much more frequent, and there's many more different types in things like brain metastases relative to gliomas. So here's all the different types of immune interactions you can have. You can have, sort of singles, a T cell interacting with a monocyte. And notice the nucleus here is not phospho-STAT3 positive. It can be an immunosuppressive interaction. Here's a T cell interacting with a CD163, M2 macrophage. Look at the nucleus here. It's green as can be. You can have multiple, as I was telling you, sort of these big cluster interactions. Here's some CD3 T cells interacting with this complex antigen presenting cell that hasn't been described except one time before in the literature. There's a great deal of complexity, and the multiplex that I just showed you, that's very, very simple. Think about what this is going to look like when I add in B cells that also are capable of antigen presenting cells. What would it look like when I add in NK cells, when I start adding in effector responses? So the bioinformatics on this has really been a key initiative for us moving forward on how you analyze it. But this just sort of summarizes the immune interactome. You don't see these interactions in necrosis areas of GBM. These are the types of interactions you see in gliomas. There's significant differences between gliomas and brain metastases. And why is this important? Well, I was taught this. I was taught you have a tumor in your head. The antigens drain down to the cervical lymph nodes. Dendritic cells take up those antigens, present them to T cells. The T cells scamper up to the tumor microenvironment and then exert an effector response. I suspect that's the way many people think about this. But this is not what the science is showing us. the science is starting to show us that there needs to be, or there is a critical event that happens in the tumor microenvironment where the T cell is re-engaging antigen presenting cells in the tumor microenvironment. And I suspect this is probably important because if the T cell is not yet exhausted during that activation event, then those T cells may be responsive to immune checkpoint inhibitors. So coming back to this, I talked to you about this side, the STING, and I'm going to finish with the STAT3. So the STAT3 inhibitor we developed was about 15 years ago. It is a small molecule inhibitor. It is highly blood-brain barrier penetrant. It can achieve very high concentrations in non-tumor bearing mice. It blocks the translocation of the STAT3 complex, the dimer, into the nucleus. As a monotherapy, it's been shown to work against a wide range of malignancies. But intriguingly, it's not particularly great against GL261, that particular mouse model. So what we did was we actually took 60 milligram per kilogram. That's the dose we use in this particular model. And we looked at evaluating it in combination with a radiation. And a radiation can draw in immune cells into the tumor microenvironment. And there's also some data about the fact of whether it can actually activate and sort of trigger dendritic cells in the tumor microenvironment. So we used 2 grays, which is what's used in clinical practice. And you can see that there's an increase in survival, the sustained survival, when you use this combination. This is solely immunological. This is not a direct effect on the tumor because if you do this exact same experiment in nude mice that do not have immune cells, there's no therapeutic effect. So there's immune memory that is induced with this particular combination when you re-challenge. So we went in and looked at the tumor microenvironment. And this is a window of opportunity analysis now in mice. So we took the mice, we treated them, and during the therapeutic window, we analyzed the tumor microenvironment. We did that in a variety of ways. We did it genetically with nanostring analysis, and we also did it with immunohistochemistry, and we did it with flow. I'm only going to show you sort of a couple of the data. So with the immunohistochemistry, dendritic cells are green, T cells are red. You can see that there's increasing interactions here. There's cluster interactions. And in fact, only those cluster interactions happen with the combination. So you do not intrinsically get these sort of dendritic cell, T cell clusters unless you give both drugs. You give the radiation and you give the WP1066. When we profiled the dendritic cell function, this was markedly increased in the tumor microenvironment. So I'm not analyzing cervical lymph nodes or peripheral dendritic cell function. I'm specifically monitoring it in the tumor microenvironment. So this is what the drug looks like. It's an oral drug. It's coated with methacellulose. This is how it's administered to patients. They get this box. This is the vial with the drug, and it's covered. They take this little vial that has a flavor. They mix it, and then they shake it up, and then they swallow it. And then they take the second little vial, and this is to clean out the first little vial of any little particles, and then they take that second shot. And so this is, so it's BID dosing. So this would be a day's worth here of the drug. And then it's given 2 weeks on, 2 weeks off. And that was worked out with the mice. So we've completed a phase 1 clinical trial. We got to a dose of 8 milligrams per kilogram. And patients started experiencing the aspirin-y taste that they didn't want to take more of. So it was not a maximum tolerated dose, but it was a maximum feasible dose for patients. Allometric scaling of 60 milligrams per kilogram, which is what I showed you in this experiment here. The equivalent in human is 5 milligrams per kilogram. So this clinical trial exceeds the dose necessary for the combination with the radiation. And so this is typical of what you would see in a phase 1 clinical trial in a small group of subjects. We did not see a meaningful radiographic responses in GBM patients. However, we have this trial open at Emory with Dr. McDonald, Toby McDonald, and he has had 2 meaningful radiographic, 50% radiographic responses in 2 DIPG subjects that have been treated with WP1066 as a monotherapy. And there's some reasons for that biologically, potentially. You can see suppression of phospho-STAT3 in the peripheral blood. And then ultimately, here's what the phase 2 clinical trial is going to be looking like moving forward. So we're going to be going in the newly diagnosed MGMT unmethylated patient cohort. These will be patients receiving radiation plus the drug. We then will be monitoring them for response, i.e. progression-free survival. And then additionally, we will have a window of opportunity cohort for those patients that did not, let's say, get a good resection, et cetera, only got a biopsy, in which we take them for optional surgery and then investigate the tumor microenvironment for the induction of cluster reactions with the combination. So key take-home points, there's different mechanisms of immunological responses in GBM that we may not necessarily be able to use the same sort of therapeutics other folks use in the oncology community. Immune therapeutics may not be working the way we think they might be. We need to be thinking more about how to bring T-cell responses in the tumor. I think that really is the neurosurgeons here in this audience. We need to think about a little bit more about how we classify monocytes, macrophages, microglia. That's for our basic scientists in the audience. And I think increasingly we're going to start thinking about therapeutics that modify immune cell interactions as far as the next generation. And so I want to leave some time for the audience to ask some questions and for me to take a sip of my coffee.
Any questions?
Thanks, Dr. Heimberger. That was a great talk. I think for the neurosurgeons, I guess, in the audience here, curious in terms of delivery approaches, in terms of giving these targeted therapeutics or even CAR T cells. I know there's a number of papers that just came out, one, I guess, in Nature by the Stanford group, kind of showing that if you give CAR T cells intratumorally or in the CSF, they actually have much more a higher effect than, giving an IV. But I'm curious if the same kind of I'm guessing you're seeing the same kind of results with your targeted therapeutics as well.
Yes. So again, with the cellular therapies, a lot of them, again, this sort of immune topographical atlas that we've started working on. I mean, it really does show you where the T cells kind of hang out. So this is where you'd expect an IV administration strategy to sort of be in this perivascular space and that kind of be hindered in that manner. So, I think that ultimately, with intratumoral injections, you're going to be limited. So we really have to start thinking about how we're going to do that as neurosurgeons for our oncology community. Now, CARs are obviously challenging for a variety of other reasons as well, which is we don't have a lot of great antigens in the GBM domain. Most of those antigens are heterogeneous, and we've done a number of clinical trials that have shown that any degree of heterogeneity essentially leads for, selection of, escape variants and, subsequent progression. So the IL-13 RA2 illustrated that, the EGFR-V3 CAR T cell illustrated that. So, I'm so far not convinced that there is a uniform antigen that we can go after in GBM. Now, I have a different opinion for other types of tumors within the central nervous system that may be much more clonotypic, but I think that's one of the key limitations for CARs, in addition to this distribution issue that, is becoming more and more obvious through multiple investigators.
And then my second question was, I guess, knowing that, you were closely with Adam Sonnabend, is this kind of focused ultrasound window that they're kind of using from the European group in early phase clinical trials, if you're finding any improvement in drug distribution using that kind of model? I know you used it in mice. I saw it from the cranial window. But...
So certainly I will leave it to Dr. Sonnabend and he's a wonderful speaker, by the way. The device that we use was the one that was initially described by Alexander Carpentier. And so there's a Science Translational Medicine paper, I think it's from 2016, it could be 2017, where he describes a single array that delivers ultrasound to a very small sort of, I would say, cone of opening. And so that was about 1 centimeter cube as far as sort of that cylinder that he was able to open. The newer devices is an array of 9. And so you have a much greater distribution area. So the device that Adam is using is one that you actually implant in the skull. And so that 9 array is kind of malleable. So obviously it's more challenging to place one of these devices if you're talking about posterior fossa GBM. So it really kind of has to be, sort of in a good location where you can sort of position it with a Calvera. There's limits and advantages of, of everything. And so he is seeing increases in concentrations. There's some caveats to that. And I'm not so sure that it's my story per se to tell everyone, because that's really his science. And I don't want to usurp any of his sort of, how do I say that, presentations. But yeah, there is an increase in concentration of drugs that he's using in areas of the blood-brain barrier opening, but it's not uniform. So there's probably some additional tweaks that need to be made here.
Dr. Heimberger, great talk. I mean, amazing work that you're doing, and we congratulate you and thanks for your time today. I just want to know your general thoughts on, after we kind of get to a point now where we're kind of getting these really great cellular kind of results. What else in the patient's environment can we optimize to kind of help with these responses? Things like diet, Decadron, that they're kind of getting before and after surgery that could also play a large effect on their immune status.
So, David Reardon has been a huge advocate for, minimizing steroids, and the data is quite clear, for other cancer lineages that the use of steroids, really does sort of inhibit immune checkpoint therapeutic effects. So, certainly minimizing that. I don't know if we can go as far as whether there would be acceptance in completely stopping it. I don't think that there's some clinical indications. I just don't think that's going to be feasible. But certainly there's sometimes some folks that have a right frontal tumor. Maybe it's very asymptomatic. Maybe we could really curtail, the use of steroids in those patients. But there's so many other factors besides the iatrogenic induction of things like temozolomide, chemotherapy, all of those kinds of things. There's just such a profound immunosuppression in of itself of the tumor. Certainly they're not helping. So we can certainly control those things that we give patients. Did I answer your question? Or was
Yes,
I feel like there was a second component to that.
Diet was the other thing, people.
Oh, diet.
People
Okay.
are looking at like gut microbiome and kind of the effect on tumor, and I just feel like that eventually is going to also play into the immune status as well.
I don't know enough about the microbiome to really comment on that. Although I will tell you, Jennifer Wargrove, about 10 years ago, we were quite interested in looking at that relative to GBM patients. I don't know if it has an influence in outcome. There has been a fairly robust discussion, including from our group with regards to the ketogenic diet. There's groups that say it works. There's other groups that don't say, it works. I think if it doesn't impact patients, to the point where they really feel like it's, something negative in their life, I don't have any objections to the use of it, is typically how I always phrase it to patients. But I'm not aware of anything really profound immunologically that it modifies, when you do it. Craig Korbinski published a fairly negative study at PLOS-ONE about 4 months ago on this, where he kind of did a tremendous amount of due diligence on it to kind of negate that he really couldn't see much going on. I do, there are some interesting things that are starting to emerge with regards to IDH1 inhibitors and their immunological roles. I think those might be interesting as far as combinatorial strategies moving forward, but that's not a diet-related question. It's more of a metabolic. As far as other sort of, dietary modifications of the immune responses, I'm not aware of anything that really has really significantly, compelling, data.
I know there's a bunch of people on Zoom. Anybody else have any questions?
Hi, Dr. Heimberger. I'm one of the neurosurgery residents here, Sumed Shah. I just wanted to ask a quick question about, you had mentioned the perivascular niche in relation to the macrophage, like the immunosuppressive macrophage kind of environment that exists in that area. Have you looked at what the focus ultrasound does in that perivascular niche after ultrasound use in order to break the blood-brain barrier there? Does that change the macrophage, I guess, like concentration in that area?
I have not looked, but I think that's an interesting question. My guess would be, if you're saying, what do you think scientifically is going to happen? I suspect it's just a matter of it separates. They're probably not going to go away. I bet you those CD163 are so deeply embedded, but that would be a cool study to do.
I had spent some time with Manish Aghi over at UCSF looking at perivascular neutrophils in that area as well. So I just thought it was a very cool slide. It took me like 2 months to get a figure like that. So congratulations.
So with Manish, and he does some really wonderful work. Are you guys, I'm just curious, have you guys like seen some macrophage and neutrophil interactions with your analysis?
That's a really good question.
Increasingly, so in other cancers, that may be the case.
Yeah, so the things that we started seeing was that the neutrophil microenvironment in that area can relate with and somewhat influence macrophage polarization, and actually vice versa. We looked at neutrophil polarization in that area and found a protumoral neutrophil phenotype in that area as it related to GBM stem cells. So that's kind of what our kind of focus was. It was an interesting year for sure.
Yeah, and Manish is a wonderful mentor. I have only amazing positive things to say about Manish. You were very fortunate to work with him.
Absolutely.
Do we have any other questions? Or Zoom listeners? Do we have any question from Zoom? Any other questions?
Everybody's trying to get their coffee in.
Doctor, if you want, you can start your other talk.
Okay, you've
I
really
don't think we have any more
gotten
questions
working
for
today.
now. So you can go ahead, share, and we can start whenever you want.
Okay, I just had to load another.
Thank
thing.
you.
Okay. So I kind of got the impression that you guys wanted me to speak a little bit about the surgeon scientist. I'd really love it if you guys jumped in the middle of this as opposed to me just giving a lecture. I think many people aspire to this, and certainly, a lot of programs really sort of push this pretty hard. But I think it's something you have to decide for yourself, obviously. And my talk's really directed in many ways to the residents. So, although this is touted as sort of a goal, I will let you know that this is kind of a minority population. So, this study was done about, I'd say 6-7 years ago, and this is courtesy of Linda Liao, who's chair at UCLA. And the analysis was done looking at the total number of RO1 awards. And you'll notice that this is not particularly high when she did this analysis. There's about a total of 90 plus neurosurgeons across the country that have submitted applications when she did the analysis. And the success rate is fairly good. But overall, if you look at the number of PIs with active RO1s, the number is, kind of less than 1%. Some of the reason these numbers look a little sort of odd in some ways is the fact that, a lot of neurosurgeons, if they get one, many of them have more than one. So that's how come you're sort of seeing that number go down, is because in order to run a lab, you usually need at least a couple if you're really running, a fairly vigorous research program. So one of the things that folks ask is, what are the keys to the success? And I think one of the key elements is actually mentorship. And there's a difference between mentorship and sponsorship. Mentorship is someone who's really helping you sort of with an idea or a research project or a concept or a clinical trial. A sponsor, in many instances, is a more senior person. Doesn't necessarily have to be. Sometimes you can have a junior faculty member who's a great sponsor, but a sponsor is someone who really recommends you for things or helps get you on committees or get you appointed to stuff, and kind of helps you politically move from your, let's say, just exclusively your institution to a more national scale. One of the keys for mentorship, though, is not a single mentor. And in fact, I continue to have mentors, even, you know, at this sort of mid-career stage that I'm at. And I suspect I continually will have mentors until I eventually retire. These mentors can be within different contexts. So when I was training, I had multiple scientific mentors. Some of them were very directed at basic science. Some of my mentors were based in like a different area that I was trying to learn. And then I also had clinical mentors. And there's been many, many, many studies that have stated that your success is really a determinant of how well you can navigate mentorship. There's different types of mentoring relationships, just so you know, there's not just, you know, one sort of the traditional way. The traditional way, I think most people think of this as one-to-one, you know, where you have a more senior faculty member and you meet and talk about things. And you know, certainly there can be a lot of satisfaction, but if that mentor is very limited with their bandwidth, their ability to meet with you, et cetera, and then obviously you don't have, you know, a ready access at hand. And additionally, you're only getting their perspective as opposed to many different sort of perspectives on how to deal with the problem. You can also have peer mentoring. So a colleague can also be mentoring you in a particular area. And there's been a couple of times I've done that. Where I've mentored co-faculty on other grants. You can have collaborative peer mentoring, where it's a group of you together, facilitated. Sometimes I have junior faculty, we get together in a group and they're mentoring somebody, let's say in the lab, and then I sort of just oversee sort of like the committee chair in that kind of context. And then you can have a variety of different types of combinations of these kinds of mentoring. One of the things when finding mentors, I think it usually works out best when it arises organically and the mentee actually identifies the mentor. There's a number of characteristics to be looking for. You really kind of need to find someone that's honest, mature, balanced. If you're looking for a mentor for research, it's very important that you find someone that is regularly publishing. One of the issues I had to deal with about 10 years ago was I had a candidate who was working on his PhD thesis, and his mentor was a faculty member, and this faculty member only published, let's say, once every 5 years, and it had to be in one of what I call the CNS journals, Cell, Nature, or Science. And so ultimately, this young man never got his publication out because his mentor would never agree that the quality of the science was sufficient in his mind for it to be submitted. And so you can do due diligence on your mentors through either PubMed, Scopus, et cetera. There's many ways you can look at making sure that this person is productive. They also know the journals usually pretty well and what the journals may be looking for. For research mentors, it's good to find someone who has got fairly well funded. This means the person can help you and guide you in this aspect of your training as well. They need to be able to provide you with insights. I don't think a good mentor should necessarily solve your problem, but they should help facilitate how you think about it. Because if they're always solving every problem for you, that doesn't enable your problem solving skills. Screening tools for selection of a mentor. Go to PubMed, go to Scopus. I actually recommend you attend their talks. I do want to make a comment about this. You do have to be a little bit wary of what I call the cult of the great presentation. Occasionally what happens is you'll be at a national meeting and someone will give this absolutely amazing talk, and you could become very enamored with the science or with the personality of that person. That person may or may not necessarily make a great mentor. And so you need to actually go to multiple talks, interview them, talk to them before making a commitment that this is the person you want to spend time with and invest your precious research time working alongside. I always recommend you do a background check. In other words, go talk to their other mentees, understand where their mentees have ended up. If their mentees have ended up in industry and you don't want to do that, then that might not be the right mentor for you. I highly recommend attending their live meeting. These are usually the people you're going to work with. And additionally, it helps you understand the dynamics. So folks are usually asking whether they should get someone experienced or whether it should be a more junior faculty. There's a number of advantages and disadvantages to both. An experienced mentor is capable of also acting in a sponsor capacity. They usually have very well-established track records. They a lot of times have very good political connections. They also understand what a given journal may be looking for. I also serve as a deputy editor for JCI, Journal of Clinical Investigation. And so I have a pretty robust understanding of looking at the abstract and the figures of whether that particular manuscript is going to meet muster of even being sent out for review. And so more senior faculty can help guide and prevent you from wasting time at journals where it's unlikely that it's going to fly. They also have a very extensive knowledge of funding sources, various sort of venues, not just the NIH, but also the now foundations. And as clinicians, you also have the fantastic opportunity of philanthropy and grateful patients. And so that really is extremely helpful to you as a surgeon scientist. The experienced mentor also has extensive contacts that can help you facilitate interactions throughout your career growth, promotion, etc. The downside to an experienced mentor is they may or may not necessarily be up on the most contemporaneous methodologies, and they may not necessarily be able to appropriately advise you with the transition into faculty. So, 20, 30 years ago, this was how, the labs got set up, and that may not necessarily be applicable, nowadays. So a junior faculty has recently navigated that transition. And so they might be much more informed as far as these processes and sort of, navigating transitions. A lot of times they're much more closely invested and involved in the experimental design. So if you feel like you need more of a partnership alongside someone to help you work on sort of how to develop these assays or how to design your experiment, a junior faculty may be able to spend more time with you. They may also be much more invested in your success. I've actually had experiences in labs where the same project would be given to two individuals, and who actually accomplished it was the person who got the paper. Now that kind of thing now is very much frowned upon, but that certainly happened, back in the day in science. So senior investigators usually have many other responsibilities. They may be a chair, they may be on a tremendous amount of committee. They may not necessarily be able to, do a day-to-day sort of supervision of experiments. You may not necessarily want that. So it's good to gauge, what are my needs? In other words, have I already done a PhD? Do I just need to interact with someone on, a weekly or, bi-monthly kind of basis with regards to these experiments? Or am I just starting out in research, and do I need a more intensive experience? And you need to gauge that relative to what your needs are and whether that mentor can meet that. So with regards to characteristics of successful mentoring relationships, I suspect there are some people here in the audience that have had negative mentorship experiences. Every time I speak to residents, I always get some kind of story from somebody, usually over dinner. But usually the characteristics of success are, in other words, a mutually respectful interaction, setting clear expectational, personal connection, shared values. Usually where these go off the rails is poor communication or limitation of the mentor as far as their ability to interact, lack of commitment. Maybe there's just too much going on. I do tell people no if I really don't think I have the bandwidth to actually be able to facilitate them. But some people do feel bad about telling somebody that potentially wants to come into their lab no. Personality differences. I've seen this play out in a number of different ways. Perceived or real competition. I'm very much aware of multiple scenarios where a grant's been put in by a mentor, both as an R and a K, and both of them have been scored well. And then the question is, okay, then who gets the award, because you both can't. So you kind of need to negotiate that. If that's a situation you find yourself in, what will be the scenario? Will my mentor forego his or her R01 so that I can take the K? Or do I have to forego the K? So it's good to have those discussions up front before it actually happens. And then sometimes mentoring relationships fall off the rails because the mentors just don't have that experience. So one of the key things I think that surprises folks is that a mentor may assign or bring up a research project, and even though they're faculty, they may not have done due diligence. And ultimately, if you are the person who's running that project or the project leader, it is your responsibility, even if you're the mentee, to actually do the due diligence. And background reading is absolutely essential, because you may get 6 months into the project and find out it's already been published. And if it's already been published, the fact that it's a confirmatory study means you're spending an awful lot of time and effort on the fact that it's probably not going to be a very high impact or well-cited manuscript. So don't assume your mentor has done due diligence. When someone really has a couple R1s going, they've got many different projects going simultaneously, and it's really not possible for a PI to really know in depth every single project and keep an eye on the literature on everything. One of the things I've seen at mentoring PhD students is the fact that they derail their hypothesis. In other words, they have a clearly articulated hypothesis up front, but then they start getting some data that they find very sexy, and then they decide that they want to explore that. And so what they've got is 3 or 4 pages as they're finishing, or 3 or 4 manuscripts, I should say, as they're finishing their thesis, going in 3 different directions. So you need to keep focused on whatever your study hypothesis is and see it to the end. You can modify the hypothesis if you think that that's a little bit better or more salient or more important, et cetera, as you go along. But you need to be careful about the divergence of hypotheses, because it's very hard to sort of consolidate different disparate hypotheses into one manuscript. This has probably been said, especially by the faculty there at the University of Miami. Your record is really viewed with regards to the quality and impact of science with regards to science. If you're thinking about a K or a 99, it is important to realize that as a study section reviewer, chair, etc., we do not look at your case reports or your case series if they're not related to the scientific hypotheses. They don't count. It's only the research that is aligned with the scientific hypothesis at hand that counts towards your sort of expertise or your developing expertise in that domain. So it's probably better to do one really good paper as opposed to dozens of little things that nobody really reads or recognizes. This is something folks do not do very frequently, which is at the very beginning, engage the statistician. And this needs to be a continuous process. And I will tell you, I have my statisticians on speed dial, and I talk to them almost on a weekly basis. And the reason is increasingly journals are very focused on the appropriate statistical strategy, and doing it after the fact, you might find your studies are underpowered, and you will be called to task by this by the editors. In other words, you may have to actually do additional experiments because the statistical method you apply is not going to be accepted by that journal. So I know sometimes at the very end of everything, somebody throws some student t-tests on it. That increasingly is not going to be sufficient. I would always make sure that the mentor and other people that are involved in the manuscripts are meeting on a regular basis. And I will tell you, one of the strategies that I have found to be very successful is when we think a manuscript is about 50% done, we do a manuscript meeting where we bring together all of sort of the co-authors. And then we actually look at data in hand and clarify what are the experiments that we think we need to do to sort of fill in and address the remaining sort of key elements of the hypothesis. This prevents this sort of at the end where you're having to do dozens upon dozens of experiments in order to finish the paper. I will tell you that even if you've been a PhD student, usually the first couple months in the lab, when you get into it, it's not going to be particularly productive. You'll forget controls for whatever crazy reason. It really doesn't happen as far as golden research until it seems like the end of your rotation. It's just an unknown phenomenon of how it seems to happen. Reasons for conducting research. There can be multiple reasons why people do it. I will tell you that the reason I've done it all this time and the reason I find personal satisfaction in doing research is sort of being the sort of the first to know something. or the thrill of the actual discovery. But certainly, there's other reasons for conducting research, such as gaining sort of an understanding of the process, being able to look at other people's research with a critical eye. Maybe there's just a very specific question you're really interested in addressing. So many times junior faculty and residents have a hard time kind of coming up with ideas for a research project. You may have an enfolded research fellowship or research time as part of your residency. There's a number of ways you can sort of find these ideas. A lot of times it could be a discussion between clinicians, which way is better. That's where a potential area of investigation can come from. You may be making assumptions in the OR or assumptions in science. The question is that really the case? You could actually sort of think about things as far as this is a real problem that I think needs to be addressed in the field. The other way to find ideas, and this is cheating a little bit, but it's certainly a way you could do it, is going to thought pieces or review articles and looking at future directions, because sometimes they'll actually help you. And sorry about that. The residents are calling me down here. One of the ways you can sort of do that is look at where the future directions are, or areas or gaps in knowledge. And review articles can also help you sort of find areas from thought leaders that are possibly important. So how to choose the topic? I would suggest you pick something that is of interest to you and that you're passionate about. Don't choose something just because of the cult of the mentor is what I call. In other words, the mentor is excited about it, so you secondarily become excited about it. What are you personally interested in? The other thing I think that's really helpful is to identify research projects that align with your clinical interests. I'm not sure it makes much sense to, I had someone show up in my office that was a spine surgeon that wanted to do glioblastoma research. I'm not saying it can't be done, but it's a lot easier if what you're doing clinically actually complements what you're doing in the lab. And so what you discover in the lab, you implement in clinic, and then whatever you're doing in the clinic or in the OR, you actually then send to your lab. So that's very synergistic, and I think it's fairly effective. So one of the keys is reviewing the literature, and I don't mean review the review articles. I have seen in many instances where a false narrative has been propagated through multiple review articles over a series of years. And I think some of the senior faculty, like Dr. Harris, has probably been very familiar with this as well, where if you go back to the original report, that's not correct. And so I really strongly recommend that you actually sit down and pull the original literature and look at it with a critical eye. Look at the abstracts and look at the actual figures and the data. Obviously, I think everybody here knows you can PubMed, Google, there's many different types of venues for looking at the literature. So there's levels of evidence, just so everybody is aware. 1A is the highest. These are meta-analysis of randomized controlled trials. A lot of times your National Comprehensive Cancer Network, as far as making recommendations, those recommendations are based on the level of evidence. So yes, you do need to know this information. Grades of recommendation. A's, those include your randomized controlled trials that have narrow confidence intervals. And then less are sort of your systematic reviews of cohort studies. And then obviously your least sort of level of evidence is an expert opinion as far as what would be accepted when you start thinking about alteration of standards of care. So formulation of a question. So when you start with a hypothesis, you have to start thinking about how you're actually going to address that hypothesis. And 3 key things to think about is, how well grounded is that question in the actual knowledge base? Can you actually research it? And how important is it? So don't waste your time on an incremental small analysis. If you're really trying to move forward as a surgeon scientist and you really want to have impact, go after the bigger questions. But it can't be so big that you can't actually accomplish the study. So a hypothesis, by its definition, is there must be risk. It can't be something that it's an observation. It has to be something you can falsify. In other words, it can a null hypothesis. You have to be able to negate it or refute it. So how do you actually go about doing a hypothesis? Well, you have to have your research hypothesis, and then in combination, I cannot emphasize this enough, you must have a statistical hypothesis. So when I have someone putting together a research project, I make them write the introduction, and I make them tell me the hypothesis, both the research and statistical. Now, they have to do this before they actually do the research project. Why? Because writing that introduction forces them to do the PubMed research, and the hypothesis means very clearly, what am I really going to do? So I have found this to be extremely helpful in focusing trainings. When you start the science, and actually, how do you start, moving to the next step, which is called operationalization, then that's where you need to sort of engage the mentor. So what is operationalization? So let's say my hypothesis is this. Children who watch television become more violent. Okay, let's just say that that's the hypothesis. To operationalize that, okay, children, define children. What's the age category? Watch, what's the minimal interval of time? Watch TV, is there particular types of programs? Do they need to be particular violent programs, or can it be any kind of program? If it's a violent television program, does it involve hitting, or does it simply involve shouting? So you can see all of the steps in order to address that hypothesis that you sort of need to go through. Are we talking about when children become more violent? Is it just that they raise their voice, or does they have to hit a sibling? So what is the perimeters of actually testing that hypothesis? So that is all the steps that you do as you move forward with the research project. So designing the study, you have to think about how is the study going to be conducted? What will be studied? And I've elaborated a little bit on that with the operationalization. Where will it? Will this be done in the home? Will you bring them in and make them do it in a lab situation? Who's going to be studied? I touched on that. How is it going to be tested? Is it going to be, cross-sectional? You're going to take these kids, you're going to expose them for a couple months to violent television and then observe a behavior, and then you're going to then not expose them. Or are you going to do cross-sectional? In other words, you're going to take a group and randomize them to violent television programs versus not. So the key components of a research proposal, the thing that one looks at is it impactful? I've said that multiple times. Is there merit to this? Is it novel? Is there something unique that is really going to extend our knowledge base? Is the hypothesis valid? Is there evidence to support it? Are the aims logical? Is the procedure, i.e. this is known as the approach, are the investigators qualified? And is the facility adequate? And this is exactly how the NIH assesses research. They break it down into actually a little score sheet, which I'm going to show you, but these are really the key components of a successful research proposal. So research opportunities for neurosurgery residents. There's a number of programs that have R25s. These are specific for residents or fellows in neurosurgery, neurology, neuropathology, and or neuroradiology. These are typically applied for by a faculty. This is not something that the resident actually applies to the NIH for. It's something that the faculty does. And then it is either peer reviewed or a potential person is nominated to participate as the trainee in this program. The idea is that fosters careers as physician scientists or surgeon scientists. You do have to commit a certain amount of time to this, devoted specific to the research effort. And then there's a number of programs. This is directly off the NIH webpage, who actually has active programs, R25 programs. These may not all be necessarily neurosurgery. Some of these are exclusively in neurology for some of these programs, or specifically like neuropath, but University of Miami does have one. But you can sort of see who amongst our colleagues also has these particular mechanisms of support for the residents. For Northwestern, we have one, but it's for our neurology residents. But there's other funding sources for our neurosurgery residents if they choose to do research outside of the R25 program. So grants for junior faculty, there used to be a lot more. There used to be K awards. Now the two that mainly are considered are the K award and the 99 and 00 mechanism. The K award is mentored. Part of the that grant that's important is who you pick as a mentor. So that person needs to be particularly strong and viewed as a good mentor by the NIH. And it comes back to some of the criteria that I previously mentioned. This needs strong institutional support. You can't have your chair just say, yeah, we're giving you 50% protected time. It really has to be carefully delineated in a chair letter that, okay, I'm on clinic on Monday and Tuesday and I operate or whatever. And this is, they really do need to see that 50% or greater commitment to the research in order to be eligible. And usually you need to apply within the first 3 years of the faculty appointment. So K awards, those that have received K awards in the past, typically you do have a pretty good chance that you're going to get an R01. It does take time though, from when you first get your first K award and move to your first R. Usually the average is about 6.3 years on average. 99 to 00. This is really fairly well suited for folks that have a PhD or significant funding under their belt or significant research under the belt. The first 2 years are mentored research. That's the 99 component. And then the 00 is then as you transition into faculty. The challenge with this particular grant is if you move institutions. In other words, if let's say you're a fellow and you're doing the 99 and then you move to the 00 and some, sort of your infrastructure and sort of your collaborations sometimes can be a little bit of a challenge for this particular mechanism. So strategies for grants. One of the key things I've run across, and again, some of the senior faculty members I'm sure are very much aware of this, but a lot of times the trainees are not. If it requires an institutional signature, that means it needs to go through typically your institutional grants and contracts office and sign off. And almost none of the institutions I've ever heard of do it in a day. Most institutions that have high volumes of grants are going to require you get it in 2 weeks before the deadline. I have actually lost grants because we can't get it through our grants and contracts office in a timely fashion to meet their criteria. So in addition to the grant timeline you see on the RFA or the request for application, there's also institutional deadlines you need to be aware of and make sure that you meet that. I strongly recommend that you get multiple people look at your grants, especially if you're sending them at the national level. This is your calling card, and a crappy grant will become known. So especially if you've put in multiple poorly written, poorly considered grants, your reputation will precede you. As a chair of a study section, I will tell you, we know the names. And so you want to make sure that you put in the best product you possibly can. And that is not just the way you think about things. Get someone senior. And do not get someone who's your friend to read the grant. Get someone who will turn it blood red and will have 100 comments, because you're either going to have to listen to these comments then before you put the grant in and try to address them, or ultimately you're probably going to hear these comments after you get the study section review comments back. If you're a mentor, don't write this for the mentee. They're not learning anything. If you have grants that are sent to multiple funding agencies, make sure you kind of have clarity with regards to which grant you're going to accept if a duplicate award is made. I touched upon that if there's a problem with overlap with your mentor. And then a fact of the life is, this is just like being an actress in Hollywood. You're not going to get every role. You're not going to get every grant. There's no one in the world that has gotten every grant they've applied for. I know people may make that claim, but trust me, when you start doing the due diligence, no one gets every grant. So you do have to put in a fair amount of grants to keep a grant fairly, or keep a lab fairly well funded. One of the tools that you can use, one of the things I've heard people have a lot of struggle with, is which study section to put the grant into. NIH has this ART tool, an assistant referral tool. This is really fun. All you have to do is put in the title and answer some questions, and essentially it will rank all of the study sections as far as alignment to match. And that's how you can sort of find it, or prioritize which study section. You need to designate which study section in your cover letter you want this to go to. If it includes a clinical trial, by mandate, it needs to go to the NCI, if you're talking about neuro-oncology. If it is preclinical, it can go to NINDS. It's better to try to get it to go to NINDS because the pay line is more, it's easier at NINDS relative to the NCI. So my suggestion would be NINDS aligned study sections. So this is how research is judged. It's a score system, 1 to 9, 9 being poor, 1 being exceptional. The goal is to try to get as many ones as humanly possible. That's the goal of the study section. This is what an RO1 sort of the scorecard looks like. You have overall impact. You have then subsections, significance, investigator, innovation, approach, and environment. Now, the overall impact may not be a median or a mean of the subsequent subsections. So someone can weight approach much higher than all the other subsections and ultimately give you, a score of a 5, even though maybe all the other sections were like ones, and then, maybe only the approach was a 5. That doesn't mean your overall was, like 2.4. It's at the discretion of the reviewer what the overall impact score is. It's their thinking of when you put everything together, where does that number come in at? Significance. This means, is it really going to change the field? Is it really going to have some impact? Investigator. I touched on that with regards to strategies, with regards to publications, focus within the domain in which you're putting the grant in. Innovation. Are you really pushing it? Or is it the same old, we've seen it a million times before? Approach, this is your scientific strategy. And almost always, this is the lowest scoring component for almost all grants. After thousands upon thousands upon thousands of these, this is usually where the score sort of has problems. But it's also the area where in many ways, it's the easiest to address. And then environment, this really is your institution. This is really about core resources, training programs, educational programs, mentors, etc., interactions, collaborations, interactions. That's where all of that sort of gets scored at. So what happens at study section is 3 people read your grant in depth. The primary usually gets 5 to 7 minutes to summarize what's in the grant. Reviewer 2 gets a couple more minutes, and then reviewer 3 only adds additional comments. There may be 30 people at study section. Only those 3 have probably really read it in any real depth. Reviewer 1 will sort of set the tone for the entire group. Everyone votes on what the ultimate score is at NIH study section. So if you have somebody who brings up a key point that's a problem for the grant, this may further drive down the score, and it may not necessarily be reflected in the initial comments. And a good sort of director will make sure that those comments, or scientific director will make sure that the comments during that subsequent discussion are conveyed to the investigator so that they understand what happened during that particular review cycle. And then there's other components as well. Is it human subjects protected? Is there inclusion of women, minorities, children, vertebrate animals? There's all these other things that the reviewers can comment on, but they're not score driving criteria. So strategies for managing your manuscripts and research project. I've already told you, write the introduction first. I write the materials and methods as the experiments are done. I like to create publication quality manuscripts as the figures and data emerge. You can use those also in your PowerPoint presentations. I edit the manuscripts as it evolves. I almost always have a working manuscript for every single project we have going in the lab. I can actually show you my hard drive if you're interested in how we organize all of this as a busy neurosurgeon. I've talked about the manuscript meetings. I have an established scientific editor I usually work with. Most bigger centers have one, or a lot of senior investigators have someone they work with to help with this kind of task. Ongoing relationship with the statistician, I've talked about this. And I actually use a vetted reference database for all my manuscripts to make things easy. And then I have a written policy for authorship, validation, and reaching consensus. And this really prevents a lot of problems moving forward. So it's very clear when we write every manuscript where every name is going and why they're going in that particular order. So despite all your best efforts, you will get rejected. There's been some really big papers. This is part of the process. It may feel like someone's making fun of your baby. It certainly does feel that way, even as you become more and more senior. But it's not personal, and it will ultimately, at some point, you're going to experience rejection on manuscripts. It's very common About 69% of papers have been rejected. Obviously, the higher impact factor you go, the more likely it's going to get rejected. A lot of times these can be salvaged. There's a number of reasons why manuscripts are rejected. A lot of times it's absence of message. For high impact journals, a lot of times it's absence of mechanism, poor originality, methodologies, etc. About 23% of these can be easily easily fixed, poor statistical analysis, unsatisfactory illustration tables, insufficient data, etc. It's hard to fix lack of novelty. That's the reason why I stressed it so much during this talk. If you get that, it's really, really a challenge to sort of fix it in the manuscript revision stage. What is rejected and what is salvageable? I've already talked to you about the fact that lack of new knowledge. This is major, and this is major methodological flaws. This is also a real reason for rejection. Sometimes these methodological strategies can be fixed, sometimes not. Organizational are fairly easy to fix. The way you frame something fairly easy to fix, etc. So strategies for presentations. I harmonize all of the formatting for all the slides we use in the laboratory so that you can use them as modules between and going back. I catalog all my talks, and these are available for all my trainees, usually 1 slide per minute. We do dry runs of all of our talks with multiple individuals, especially national meetings. We increase the meeting and venue size for presentations, especially for people that are fearful of group speaking. And then we also have our trainees participate in public speaking courses and remedial work if it's a problem for them. So finally, how to select mentees. These are for the faculty. I actually pull the CV and bio sketch and look at all of their knowledge base before I make a decision. I interview everybody. I actually do a push away technique, which is technically I tell them that they need to go and interview and look at several other faculty members, laboratories before they make any final decision with me. Obviously, maturation level is important. I determine their passion and commitment to this area. Again, making sure that it's not just a matter of the fact that they become enamored of a particular idea I've given at a national presentation. I encourage dating, and by dating, I mean I encourage them to go to other labs, spend time in other lab meetings before they actually decide their final project and where they want to sort of spend their time. And I actually call previous mentors to verify skills and activities, especially for postdocs. And then I have multiple other faculty still interview at this stage, almost everybody I bring into the lab. And then I also determine compatibility with pre-existing lab members. So I make them hang out with my other lab members before we accept people into the lab. And then I make them give a talk. And then I do, regular scheduled lab meetings. I clarify objectives of all the relationships we have. I have secure data on writing platforms. I have multiple people redundant to me, junior faculty that work with the more junior folks. And then I streamline the research directions and verify complementary time utilization. And then as far as independent investigator status, I do collaborative review of manuscripts and grants. I'm involved in a wide variety of sort of institutional committees with every step along the way, IRBs, animal approval, etc. I also am involved in human subjects research education. I involve my trainees on FDA calls when applicable, and then I try to facilitate networking for them as well. And then I do a lot of delegation. So final advice, pick a project you're really passionate about. Make sure it's important and has impact. Pick a mentor that actually has a stake in your success. You will be rejected. Do not take it personally. Apply for your own funding very early in the game and as frequently as you possibly can. This really helps you understand the process. Get input on your project frequently and often, and by people, and including people who don't like your project. Don't try to do too many projects at once. Sometimes trainees feel obligated, or a faculty member approaches you, it's not really your thing, and you feel bad about actually trying to say no about it, and then you end up with 7 projects and you get none of them done. So pick the things that you really want to do. Devise your involvement in research to suit your career. I've talked about making sure your clinical practice has some synergy potentially with your research. And then try to devise your research specialty very early in your career, if possible. So I've got, I believe, 4 minutes. So I'm going to end right there. Dr. Hiros, I know you've been around the block a lot. Maybe you have some great advice for everybody.
I am unmuted. No, I just wanted to say what an honor and a privilege it is to have you here. I hope the residents realize how important it is and what contributions you've made to neurosurgery and how pleased I should be to have you here.
Oh.
It's interesting how things have changed. And I think for the better. You know, when I began, I'm sure the residents know that I'm not a scientist at all, and they may wonder, well, how did I get RO1 grants and maintain them for about 15 years? And at that time, if you were not a good scientist like you are now, you could actually invest some of the clinical monies that we brought, by having a low salary. At that time, when I was at Harvard, the Harvard salaries were very, very limited, but yet you could use the rest of the $1 million we brought a year to have a lab and to do enough of their preliminary data that then when you apply for the grant, you had already done 50% of the project you proposed to do. So it was much easier then for clinicians to use some of the clinical monies they brought, or most of the clinical monies they brought to do research and get RO1s. So I was very interested in your comments on the study section. It hasn't changed much actually from the time I was there, and I was really impressed. And I think the residents need to know how impressive the process of review on the study sections is. That really was very impressed with that. And what I think they don't know yet, and I think that you eventually will be on the council, I'm sure, of the council of the NINDA, that there is another review level beyond the study sections, and that's a council level. And that there is a much more complicated issue, because a council is divided. About half of the people are scientists, and I don't even know how I got there, not being a scientist, but probably because my name is Latin. They just needed a Hispanic name in the council. But then the other half of the people are interest groups, like the executive director of the Multiple Sclerosis Society and the executive director of the American Heart Association and so forth. And they advocate for their own interests. And then there is another layer that comes from above at the council, and that's the political that comes either from the Congress directly, sometimes from the president. And I remember being there when AIDS, for example, became an issue. Almost any grant, even though they had a very low score, if they had to do with AIDS, the council approved them. So
yeah.
there is a layer, and I'm sure that now all these grants that have to do with the differences in between white people, Black people, Latinos, and so forth. They have high priority, and
You are correct.
but it's a very interesting process, and I think anybody now who has an RO1 grant like yourself and the other neurosurgeons have, they've really deserved it, because they couldn't do it the way I did it, just by doing the preliminary data using clinical monies. You really have to do it on the value of so and we're very lucky to have one of our colleagues now have a narrow one early on in his career. So we're very proud of him, Bowie Stark. Yeah. Thank you very much for being here. It's really an honor to have you here, Amy.
So Dr. Hiros brought up a very good point that I neglected to mention. As clinicians, it's important to have diversification of the financial portfolio for running research. And so one of the distinct advantages is grateful patients and philanthropy. And usually those are unencumbered. And so that gives you an opportunity to, a source to generate pilot data that then you can leverage for peer review funding, either through foundations and the NIH. And so I have 4 funding streams for the lab. One is industry, which is fairly substantial for us. NIH, and then we have foundations, and then we additionally have grateful patients. So I have, just like your retirement portfolio is diversified, we also have a diversified financial portfolio to support the lab initiatives.
Thank you for the great talk. This is Ashish Shah. I just had a quick question. I guess now, a lot of these neurosurgery residency programs are transitioning to a 6-1 kind of paradigm, where now we're doing 6 years of kind of clinical clinical work and then 1 year of kind of post, I guess postgraduate fellowship training now. So now people, I guess a lot of residents are electing to do the clinical work instead of doing the basic kind of, traditionally neurosurgery programs, like including ourselves, had like 2 years dedicated to doing research, or at least a year and a half. And so now I feel like it's harder to kind of breed neurosurgeon scientists in this model, whenever there's such limited time for training. And there's such a onus to kind of, publish these first author basic science manuscripts. So it's almost like if you, finish residency without these first author basic science manuscripts, it's like it's very hard to even get a grant. So I was curious how, I guess on the administrative side, is the NIH trying to, look at that differently for neurosurgeons, or is that, is kind of a problem with the system? I don't know.
So, so I'll put on my NIH council hat on this then to address part of this. The NIH in some ways views that they've kind of already given neurosurgeons a little bit of a help up in the fact that some of those 50% effort commitments, it's higher for others. So, for the research time, just to let you know. So I'm not seeing any sort of accommodation at this time at the level of, I hear your pain, but I don't hear the NIH in any way accommodating. They hear the fact that, there's a limitation of physician and scientists, but I want to sort of put a little bit of this in a little context. In order to really, in my mind, to be successful at this junction for running, a lab, you really do need to think about training as hard for that as you do for being a neurosurgeon, because 1 year is probably, just FYI, going to be insufficient to learn how to really run research rigorously, really understand advanced computational bioinformatic techniques that are now the standard for many, many labs, how to write grants, how to put this all together, develop sort of the knowledge and, sort of the politics of how to navigate this kind of thing. So in many ways, the training needs to start way before residency. So to give you a little context, I was doing research in high school. I then did research every year I was in college and published papers in college. I then took several years off, did research, published fairly high impact papers during that time, and then spent every single year in medical school in a research lab, including developing a network of some of the modern pioneers of immunology. Then I actually started writing my research proposal for the AANS research fellowship, my PGY 1 year. And so I already had a lab I was thinking about working with. I'd already met with my mentor, Daryl Bigner. I had already given him a set of specific aims. And then I was already starting pilot data even before I started that time, actually physically. So I was showing up in the lab trying to run experiments before I actually got the devoted time. Then I continued on when I was finished with, doing PGY 6 and 7. I was still returning back to the lab because it was just 2 buildings over to try to finish up work and then to write. And then as I was transitioning as a PGY 7 into my junior faculty position, I was writing my first clinical trial with the AACR and, the Clinical Trials Network. So I walked through the door at MD Anderson with a IRB ready clinical protocol. And I walked through the door already having secured substantial funding in addition to my startup. And we can certainly talk about that, and handed that essentially over on the first day I got started there. So you have to plan along the way and publish. And so, when people come to me and say, oh, I want to do research, I say, oh, so you want to be a writer? And they kind of look at me kind of quizzical. The fact of the matter is, with the exception of today, I spend almost every morning writing, usually about 2.5 hours. And so that's my process. And it may be a manuscript, maybe a review, might be an editorial commentary, might be a grant, maybe this, might be that. But it's a certain discipline you have to sort of get into in order to sort of get that trajectory up and going. So 1 year in of itself is probably insufficient. So you either got to know a little bit that you want to do it before you get underway in residency, Or there's other opportunities, like we would go to the NIH after you're finished with your residency and you participate, you know, in their fellowship there. They have a very nice oncology sort of neurosurgery fellowship. And, you know, there's folks that I can certainly connect you with if you would like to talk to them, you know, about rotating and spending some time at the level of the NIH. So it's prep, it's deep commitment, and it is a 7-day-a-week kind of job. So, you know, if you're really going to do it, you want to be running the show, so to speak. It does really involve that. The alternative is you do not have to be involved in research and so intense like this, like what I'm describing, and still have really meaningful. You may be a clinical, you know, researcher where you're running clinical trials. You can either do investigator-initiated clinical trials or working with industry or device companies. That's appropriate. You know, clearly, you know, we have many different types of devices in neurosurgery. That's a way to conduct research in a rigorous and meaningful way. Or you may actually have PhD partners. Maybe that's the person that does the heavy lifting. In other words, that's the person that puts the grants in. But I will tell you, I know it sounds a little bit of a cliche, the person who gets the money controls the trajectory. So, you know, ultimately, if there's an arbitration between you and the PhD and they want to do something X, then they're probably going to win that argument because the grant is under them. With regards to, you know, sort of how to move forward in the field and foster physician scientists, it's a challenge. It's well recognized at the level of the NIH, but it's also a challenge for the institutions and the chairs. So let me give you some just background information. So you as a neurosurgeon, I'm just going to give you a broad number. Your expectation coming on faculty as a neurosurgeon is going to be about, let's say, 750. I'm just throwing a number out there. That may or may not necessarily be the case. So what happens is then you need devoted time to actually do the research and think and be creative. And so let's say it's a 50-50 split. So now we're looking at, you know, 400, you know, roughly 375, where that is not being recouped based on your clinical practice and the RVUs and the money that you're generating. So you've really got to either generate that entire thing so that the chair or whoever you're reporting to breaks even with regards to your salary. In other words, the RVUs and what you bill for completely compensates for that 750. Or you on the other side have to really start pulling in those major grants that add to the prestige and that they dean and or however it's organized at University of Miami gets the indirects back on your RO1s and the chair gets a piece of it or whatever. And so if in their minds as administrators, what they're thinking is okay, so I need about 350, 375, but think about a basic scientist who can think about doing research full time. And if you're doing something fully basic science, you know, let's say doing some kind of, you know, genetic, you know, CRISPR knock-in, knockout, basic scientists can do that. And most of the time, those salaries are under 6 figures. So most of the time you can get a really good sort of junior level research faculty, maybe 80, 85, 90K. So you can hire 3 non-tenured track. I'm just telling you the realities. I'm not saying it's necessarily right. You can hire 3 junior research faculty, non-tenure, who can do this full time. So that's the balancing act. It's not just a level at the NIH, it's also an institutional way the whole system is sort of, you know, set up. One of the things I think though that is different and the way to leverage some of this is the fact that as a surgeon scientist, there are certain things that we do that a basic scientist cannot do. And that was part of what I was emphasizing in the talk, which was you're not going to get a basic scientist who's going to provide, you know, in block resections showing you how the microenvironment, you know, transitions between, you know, different zones of the tumor. You're not going to get a basic scientist who's necessarily going to have tremendous interest in blood-brain barrier opening ultrasound, although they might play with it on the preclinical side a little bit, but really it's going to be about translation. And that's really our strength as physician or surgeon scientists is we understand how that can be implemented for a patient. So I think really the sweet spot for a surgeon scientist or physician scientist is in the domain of translational research because they understand what it means for a patient or how this would look in the context of a clinical trial for a patient. So I'll stop there.
Thank you. Thank you. I think that's very helpful. Obviously we're trying our hardest to kind of motivate our residents to, go into these surgeon scientist kind of pathways, especially since we have the R25. So it's good to get your insight on that. And I think a lot of them have started off early with their careers in science. But the question is keep them motivated, to continue that. I actually did the NIH fellowship myself.
Did you enjoy
postgraduate.
it?
Oh, I loved it. It was a fantastic experience from
Good.
the scientific side. So I'm really grateful for that opportunity. So I'm, we'll chat later. But thank you again.
I hope you didn't miss with the very eloquent exposition of Dr. Heimberger now that essentially she's saying it's difficult to have your cake and eat it too. And if you expect to have a $500,000 salary as a beginning neurosurgeon, it's very hard then to also want to be a valid researcher. So I heard that in between but the expectations now for salaries and starting neurosurgeons make it very difficult to develop into a real surgeon scientist. Anyhow, thank you very much, Amy.
I mean, I was so dead set on doing research. I mean, it really didn't matter to me. I will tell you, I was actually dead set on going to the NIH, but Ed Oldfield, he said, yeah, you can come, but then, how the NIH is, they're dependent on, congressional appropriations. He had to give me the unfortunate news that he couldn't afford to bring me. So, and then I got very, very fortuitous in that my chair, Ray Sawaya, really believed in physician and surgeon scientists and really believed in that investment. So it's really important to identify chairs or, whoever you're reporting to that really fundamentally believe in the importance of this role. And without that kind of support, it's going to be very, very challenging. If you've got someone who says, I need you to do 250, 300 cases a year, it's probably not going to be compatible. So it has to be just sort of the right alignment and the right background to sort of make it really, really happen. And I've been very fortunate. People have been very kind to me and very supportive.
Thank you.
I don't know if anybody else speaking, but
I think that's I think you have on the schedule, I think you may have a meeting with Dr. De La Fuente, I think at some point in time. Let me see.
Okay. I
don't know if they sent you the itinerary for that. Let me just.
Let's see here.
I'll say now, thank you, Amy.
Okay. All right. So let me see here. I'm just going to see what I have here. I have a Zoom meeting. Yes, I've got it. We're all set.
Perfect. Great.
This is a nice, it'll give me a little break to grab a little breakfast here real quick.
Awesome.
And then I'll log on and we'll start having a great conversation.
All right. Thanks again. And the residents will meet with you, I think, at 12 o'clock. So
now I am lacking a Zoom for that.
Okay, we'll send a Zoom invite.
Okay, sounds
Perfect.
good.
Thanks.
Okay, talk to you later. Everyone have a good morning.